Hi guys, welcome to EP Talks. My name is Federico and I'm the scientific advisor for IB USA. In this brief weekly podcast, we're going to look into interesting and applied research paper in the field of phenology. So wherever you are at work or at home, just sit down, relax, have a glass of wine and follow me. Hey everyone, welcome to this new episode of IB Talk. This week, we're going to talk about how mannoprotein influence the aging of two red wine varieties. The article is titled Aging of Aglianico and Sangiovese wine on mannoproteins, Effect on Astringency and Color, and has been published in the Food Science and Technology Journal. Mannoproteins are naturally present in wine because they are an integral constituent of the outer layer of a yeast cell wall. Mannoprotein belongs to three different typologies depending on how they are bind to the other constituent of the cell wall. They can be non-covalently bound, they can be attached to the beta-glucan of the cell wall, or they can be attached to protein protruding out of the beta-glucan of the cell wall. They can be released during alcoholic fermentation or during the autolysis process happening during aging, and they can contribute to the organoleptic and physiochemical properties of the wine. The aging on yeast lees post-fermentation can be carried out in barrel, tank, and bottles, and involve a simultaneous process of extraction of yeast component and absorption of grape constituent. Yeast cell wall products could ensure the proteic and tartaric stability to white wine and show beneficial effect also in red wines, contributing to color stabilities, to tasting improvements, and flavor complexity. Contradictory results may depend on the typology of mannoprotein use, on the timing, and on the dose. Nothing is available about changes in astringency subqualities used to describe this complex tactile sensation that is astringency perception. This study was carried out to attempt if different mannoproteins are able to reduce and or modulate the astringency characteristic during the bottle aging of wine. Aglianico wine is well known to be a wine rich in tannins and highly astringent. Sangiovese is a wine rich in low and high molecular proanthocyanin tannins, contributing to astringency as well to bitter sensation. The experiment tested pure mannoprotein, heavily glycosated mannoprotein, and a mix of mannoprotein peptides and gamma arabic at a dosage of 20 grams per hectoliter 9 months after vinification. The aging was monitored at 3, 6, and 12 months after the addition, checking for color and sensory evaluation. The initial astringency for Aglianico was around 6 and did not significantly change until 12 months, when the control reached a value of 5 and the MP treatment was the lowest at 4. Sangiovese initially astringency intensity instead was around 4.2, and after 12 months drastically decreased for both control and treated wines. No difference in astringency were perceived between them. In Aglianico, most of the treatment cluster with sensory descriptors such as aggressive, adhesive, and hard. However, the blend of mannoprotein and gamma arabic and peptides after 6 and 12 months was associated with silk, soft, full body, and other mouth cough terms, which means was related with more body. The third group, represented by the pure mannoprotein, was felt as mouth coat, velvet, rich and persistent. subqualities more related to the aroma revelation. All treatments significantly decrease the herbaceous aroma respect to the control wine. The MS treatment enhance the floral aromas, and together with MF treatments enhance the spicy one related to black pepper. As regard to odor, the black pepper was enhanced in the MP treated wine, while the balsamic note increased both in MP and MF treatments. Control wines resulted green, hard, and aggressive. That means a strong astringency associated to sourness and bitterness. The control age for 12 months was similar to wines at 3 months treated with mannoproteins. This means that the aging of Sangiovese wine for 3 months with mannoprotein gave the same subqualities as an aging period of 1 year in bottle. The last group included all the other mannoprotein treatments, and the descriptors were velvet, full body, soft, rich, and persistent. Aromatically, the mannoprotein treatment was highly effective in decreasing the bitter perception and increased the fruity aroma related to the red cherry. The floral aroma was significantly enhanced by MP and MF treatment. The herbaceous odor decreased after all the mannoprotein treatment, indicating a fanning effect was obtained on these negative molecules. The mannoprotein treatments didn't change the color of Aglianico, which was already high in color intensity and high in anthocyanin content. For Sangiovese, instead, characterized by a low color intensity and a high hue, a formation of pigmented pallor associated with color stability was monitored. The mannoprotein also reduced the astringency of the wine, and they reduced the bitterness of the wine. However, they had no influence on the polymerization of the tannins. In conclusion, commercial mannoproteins improve the sensorial characteristic related to body and persistence of wine, reducing astringency and bitterness and encouraging aroma revelation and odor complexity, while the not treated wine remain astringent, hard, and characterized by green tannins. As regard as the effect on the color, the mannoprotein were mostly effective on Sangiovese's wine, which is characterized by low intensity and high hue values, promoting the pigmented polymer formation and therefore contributing to a better stability of wine color during time. That's it. Thanks for listening. Please, if you would like to see more of these videos, subscribe to our YouTube channel or newsletter on the website. And if you like more information on IB mannoprotein, on other products that can be used in wine during aging, please contact your local IB sales representative. Stay safe, stay connected. My name is Federico, and this was IB Talks Weekly. Ciao.